Hello everyone, welcome back to one more video session. I'm a bit sleepy. The time is 2:20 right now. I just completed first year PUC concepts and I'm taking second year PUC concept now. So switching, and obviously I have already drawn the two very important diagrams, particularly for your board examination. Could be asked for 5 marks. Okay, practice very well. The thing is, I'm a bit sleepy, but okay, listen to me carefully. You are not supposed to sleep. Got it? Okay, chalo. So I'm going to explain the seventh step of rDNA technology. Okay, what is the seventh step? It is the large scale production of desired product. Yes, the thing is, I have already incorporated gene of interest into cloning vector. Then I have inserted this rDNA into a host organism. Yes, so in the previous video session I have already explained about it, how exactly we can introduce this rDNA into different host organism. Yes, now the thing is, the first question is, what is the ultimate aim or goal of rDNA technology? You know that because I have already explained that we are going to ligate the gene of interest, which is taken from a specific living organism which can express a specific protein that could be really useful to humankind. Okay, so that is nothing but human welfare. So that is the reason why I am doing all this technology, correct? Okay, so this is not an easy task actually. So the question is, what is the ultimate goal of recombinant DNA technology? That is nothing but to synthesize or to produce the recombinant protein. Yes, what is this recombinant protein? It is the protein which is synthesized using rDNA technology inside a heterologous host. Okay, so we write that to synthesize recombinant protein. So this is the ultimate goal of rDNA technology. Now the question is, what is this R protein, recombinant protein? Recombinant protein is nothing but it is the product obtained from the gene of interest inside a heterogeneous or heterologous host, not heterogeneous, heterologous host. Okay, if you want, you can just write down that, very important. R protein, it is the desirable protein or product obtained from gene of interest inside a heterologous host. Got it? Okay, so that is actually recombinant protein. Now the thing is, what is exactly heterologous host? Okay, we write that. Heterologous host. It's only for your competitive. Heterologous means, see, I am introducing gene of interest into a specific host organism, right? Why? Because I know that that particular host organism is not carrying the gene of interest. So, if the host organism doesn't carry the gene of interest, then obviously it is heterologous host. Very simple thing. For example, like I have found that in a plant cell there is one gene which produces very special protein. Okay, if I introduce that protein into a human cell and I'm going to I can just modify the human cell. Okay, that means actually I am incorporating or introducing a plant character into a human being. That's it. So here let us just assume that human cells are now heterologous host. What is the meaning? That means the human cells doesn't contain or carry that gene of interest which is present in the plant cell, which is responsible for special plant character. Yes, so which is now heterologous host, human cells. Majority here we prefer Escherichia coli. Okay, there are so many things actually, features exhibited by E. coli. So that's the reason why it is a very good heterologous host. Okay, that done, understood, fine. So to produce R protein we need heterologous host. Done. Okay, now as I said that I am discussing the seventh important step of rDNA technology. Here we need to understand how exactly we can get a large scale recombinant protein from the heterologous host. How? For example, we know that rDNA technology is done in vitro, that is in the laboratory condition, in a small scale, like take a petri plate, then do a competency process and everything. Now the host organism takes up this rDNA, done, everything is done now. But the ultimate goal is I need more, millions of R protein. Yes, so that is the thing. Within a petri plate they cannot produce because once actually the bacterial cells starts producing, dividing and increases its number, and ultimately it reaches the saturation point, because there is no space for further division, correct? Okay, it stops. But if I just transfer this bacterial colonies containing our rDNA into a large vessels, the name of the large vessels is fermenters or bioreactors. See the names. Fermenters or bioreactors are one and the same actually. Got it? There are so many different designs are available in bioreactors actually. Particularly the scientists prefer simple stirred tank bioreactor, or sometimes sparge stirred tank bioreactor. Okay, the reactors are required actually. Why? They actually provide all the facility to the heterologous host organism. Got it? As a result, they actually facilitates rapid multiplication of the host organism. The number increases. Got it? So as the number increases, rDNA inside the host organism also increases. As the rDNA increases, the gene of interest, which is ligated, also starts producing. That is transcription, translation, and next step is protein synthesis. That is R protein, recombinant protein synthesis. So this is actually the goal. I need to increase the rate of production of R protein, so that I have to transfer everything, the host organism, into a big vessel. Got it? Normally steel vessels, not your kitchen vessels. Okay. So the capacity of these vessels is actually 100 to 1000 liters. Okay, large vessels, huge vessels actually. Got it? Okay. Why these bioreactors or fermenters are preferred for the culturing of this heterologous host organism? Because you could see in this picture, I have a request. I have already drawn this diagram before the session itself, because it takes a lot of time. The thing is, you could see this diagram and just pause the video, or maybe you can find the same diagram in your NCERT also. I have not made any changes in the diagram, as it is drawn as it is according to NCERT, because it is asked in competitive examination. So just take the diagram. Okay. So if you have any confusion in the labeling part, okay. So this is one, and this is other, and this is other, because of the space available. Okay. So you could see here. This is one part. Okay, so this is. Now the question is, why bioreactors? Bioreactors actually, obviously contains lot of inlets and outlets, which actually provides us to transfer the oxygen and even the acid base for maintenance of the pH. That is very important actually. Okay, so even steam for sterilization, that is also required, because in the bioreactor, once you put the culture into this tank, there is a chance of contamination actually. So we have to maintain that aseptic conditions. That is challenging, quite challenging actually. Fine. So, okay, so even actually we need, once we put it, obviously we need to mix it every day. That is agitation. Got it? So we need to mix it. So for that, a motor is fixed here, you could see here. Okay, and then you could see the impellers. Impellers are nothing but the blades actually, very sharp blades. Okay, I think how many are there? 3 pairs. Okay, so 3 pairs. I'm talking about impeller. This is one impeller, 2, 3, 4, 5, 6, like that. Okay, which is attached to a shaft. This is the shaft actually. Okay, and this shaft is nothing but rod, iron rod. You can imagine iron rod or steel rod. I don't know. I have not seen it. Okay, so which is flat bladed impeller, which is actually fitted to this particular stirrer shaft. So it rotates. Along with that, the impeller also rotates. As a result, you could see this dot-like structures are nothing but the culture broth. What is the meaning of broth? In biology, broth is nothing but it contains the host organism, heterologous host, and oxygen, nutrients, minerals, then vitamins, okay, then whatever, let us say acid base, everything, which actually facilitates the growth of this heterologous host. So this is broth. Broth contains everything. That is the content which is present inside this vessel. That is broth. Got it? It's like liters. Just imagine 500 to 600, 700 liters. A small gap is provided for the normally, because it is nothing but fermentation. Got it? Because for the flow of the air, carbon dioxide normally, because respiration takes place inside. Okay. Now the question is as I said, why these bioreactors? I have already explained that. one more thing, what are the challenges like we face if we actually culture this particular heterologous host organism inside this vessel? The challenges could be, I think you can write down these challenges. We need to maintain this condition, that is aseptic condition number one, and we need to provide oxygen, that should be continuous. We need to provide, I think you can see like the inlets are there where we can provide. I think here I have not labeled it, it is not labeled. Maybe like this inlet could be like for supply of oxygen, continuous supply. But it depends. In bioreactors there are two types. One is aerobic bioreactors and other one is anaerobic bioreactors, because it is depending on the type of the heterologous host organism you are using in the recombinant DNA. Heterologous in the sense like as I said, some are aerobic, so we need to supply oxygen. Some are anaerobic, so that means we need to close this to avoid the supply of oxygen. Done? Simple like that. Okay, oxygen supply is very important for aerobic heterologous host. Other than that, we need to mix this particular broth every day. Mixing, agitation is required. For this we need, as I said, stirrer shaft which is attached with a flat bladed impeller. I think you can write on that. Okay, so this is actually the important thing. And other thing is we need to maintain oxygen level. That is actually one more challenging thing actually. See, you could see these are nothing but the bubbles which are created by the air. Okay, this is entirely different type of bioreactor comparatively. Here I have written simple stirred tank bioreactor. It is simple, but if you draw, it's very difficult. Okay, and even the labeling also. This is actually a bit updated version of the bioreactor, that is called sparged stirred tank bioreactor. What is the meaning of sparged? Here actually this is the impeller, okay, or sorry, shaft. Okay, so this particular shaft is actually provided. It's like a tube like with a lot of holes. That means we supply oxygen here and the oxygen now enters into this particular chamber of the vessel and they increases the bubbles. That's it. Okay, so that's what I have written here, like the bubbles dramatically increase the oxygen transfer area, or like physics students obviously know about it. Okay, so more the oxygen, it facilitates rapid growth of heterologous host organism. This is it. Okay, and what is the advantage of using fermenters? Means I have already told, like it facilitates the growth of heterologous host organism because everything can be provided. I got it? Okay. One more challenge is cooling. I forgot. See, it is nothing but metabolism itself, right? That is, chemical reactions are occurring inside the vessel, biofermenters, correct? Okay. So during that process, obviously heat will be liberated and that will make this entire vessel very hot. And that could actually kill the heterologous host also. Got it? To avoid that, we need to supply the cool water. Okay, so this is the thing actually. So supplying the cool water, that is cooling of the vessel, is very challenging. So these 4 steps that has to be done every day. Maintenance is not that easy actually. Got it? Okay, so this is actually a bit introduction about biofermenters or bioreactors. Enough actually, it's more than sufficient. What is important is you need to draw this diagram very neatly, a very important labeling part, and this one is the other one. Only the difference between this and this is here the oxygen is supplied in more quantity, that is because of the shaft, okay, which has got a lot of holes which actually releases oxygen. The oxygen is supplied continuously, but in this case at regular intervals. It could be like 1 hour, okay, now it is 2:30, now I have supplied the oxygen. Maybe like 4:30 I will supply, like that, oxygen. So it is like at regular intervals. This is continuous. Got it? I forgot one more challenge you can write down. See, because of the continuous agitation in both bioreactors, continuous agitation will be there, mixing will be there. So that could create foam. The foam. So foam is not good actually. Got it? So we need to remove the foam. So it's not easy actually to remove the foam because we cannot separate, because it contains microorganisms and nutrients. So if I try to remove, maybe like 50% or some portion of the microorganisms will be lost along with the foam while I am removing it. So it's not good actually. To avoid that, I have to put some chemicals inside. That chemical is nothing but anti-foaming agents. So I have to select anti-foaming agent in such a way that it should not cause any harm to the heterologous host organism. Done? Easy? Chalo. Okay, we will continue with one more concept, that is the continuation part of the bioreactor, that is the batch system. There are fed batch system is there, continuous batch system is there, okay, sterile batch system is also there. We'll talk about it, okay, one by one. Yes.